Hello, everyone, and welcome to today's SSCEDN webinar presented by the Simons Foundation and the Interactive Autism Network. My name is Casey White, and I'm the project manager for the Simons Simplex Collection at the Simons Foundation. We're so glad you could join us today, and we thank you for taking the time to participate in the EN community. Before we begin, I'd like to say just a couple of words about how today's webinar will work in case you've never attended an online meeting before. Today's presentation will be shown in a window on your computer, and the sound will come through your computer speakers. We are recording the webinar and will post it on the EN website so it can be viewed at a later date or sent to others who may be interested but could not attend. Please be aware that in order to minimize disruptions, all audio except the presenters will be muted for the duration of the webinar. If you speak, we will not be able to hear you. However, you can use the chat box at the left of your screen to ask questions. These can be either technical or for the presenter, and only the leaders of the webinar will be able to read them. At the end of the webinar, we'll have a few minutes for a question and answer session, which we will take directly from your questions either in the chat or sent to the email address you see on your screen right now. Please note that the speaker is not able to provide advice regarding your child specifically, but he is happy to answer other topical questions. I think that's all the webinar information I have for you today, so let me take a moment to introduce today's speaker. I'm pleased today to welcome Dr. David Amaral to the SSCEDN webinar. Dr. Amaral is a professor in the Department of Psychiatry and Behavioral Science at the University of California, Davis. He is also the founding research director of the Mind Institute, where he coordinates the Autism Phenome Project. This is a multidisciplinary effort to define biomedical characteristics of different types of autism. Most recently, Dr. Amaral has become director of Autism BrainNet. This is a collaborative effort sponsored by the Simons Foundation, Autism Speaks, and the Autism Science Foundation. I'll let him tell you a bit more about that effort later in his talk. As part of our SSCEDN webinar series, we're so pleased to have top scientists like Dr. Amaral take the time to join us and update families about the research happening in the autism field. Today, Dr. Amaral will speak to us about brain research in autism and what brain tissue has taught scientists about autism. So at this time, I'd like to go ahead and turn the webinar over to Dr. Amaral, and thank you so much again for joining us today.
Thanks very much, Casey. It's a pleasure to be here, and thank you all for joining us during this discussion where I'll try and present some of the an overview of Autism BrainNet. I really appreciate this opportunity to discuss the need for considering postmortem brain donations to advance autism research. So what I'd like to do today, before I go into the details of Autism BrainNet, is to provide some background as to why brain donations are essential for us to understand this disorder. So while autism certainly is a disorder that affects multiple body systems, there's clearly a consensus that the behavioral signs and symptoms of autism, the core features that lead to a diagnosis, due to abnormal processing of the brain. And unfortunately, at this point, it's still unclear what aspects of brain function have been altered, and the only way, in fact, that we can understand these alterations in brain function is to study the organ most impacted, which is the brain. Now, thinking about understanding alterations in the brain is really a daunting process. The human brain is really a remarkable organ. It has something on the order of 100 billion neurons and at least 100 trillion connections or synapses by which information is conveyed. And the development of the human brain is equally astounding. So the cerebral cortex, which is the surface of the brain, the folded surface of the brain that we think is probably most involved in the behaviors such as social ability that are impacted in autism, is a phenomenal developmental feat in its own right. So the cerebral cortex of the human brain has about 20 billion neurons. And the vast majority of these neurons are generated during fetal life. During the peak of neuron development or neurogenesis between the 13th and 20th week of pregnancy, neurons are generated at the remarkable pace of 2,500 per second. So when you think about the organizational feat of not only trying to generate these neurons and to have them move to where they're going to live in the mature brain and form connections, it's not surprising that there are errors in how the brain develops. What is surprising is that it ever develops normally. And generally speaking, there is normal brain development, but again, our working hypothesis is that there is some perturbation in this normal trajectory of development in the brains of individuals with autism that leads ultimately to changes in their behavior. So as I mentioned, most of the neurons in the brain are generated during fetal life, during the second trimester. And this slide that I have on the screen now shows you the course of fetal brain development, so that at 9 months, at the term of pregnancy, when the child is born, the brain does look like a mature brain. It has the folds and creases in the brain that are the sulci and gyri. But at birth, the brain is just about 25% of adult size. And even though all the neurons are there is still very dramatic growth of the brain over the first several years of life. In fact, the brain grows to about 95% of its adult size by age 6. And what's happening during this early postnatal development? Well, you can see that the overall, in this series of magnetic resonance imaging scans of human brain development, that the overall size changes pretty dramatically between a newborn at 1 week and a child that is 10 years of age. And what's happening at the cellular level during that period is that neurons are going through a remarkable maturational process. So this picture shows histological sections, microscopic sections, through the cerebral cortex of the human brain. The black dots are actually the cell bodies of neurons located in the cerebral cortex, and you can see that they're organized in 6 layers. And in the 1-month-old child, the cell dots, or the black dots, are more tightly grouped together. Their density is higher. But as the child matures on to 6 months and then 24 months, a couple of things happen. The actual black dots get bigger, so the cell bodies get larger, and the spaces in between the cell bodies increases as well. And if we look at a different kind of staining technique that actually shows us a small percentage of the neurons, what you see is that they actually look like trees. So the black dots that we saw in the last slide are the round or pyramidal cell bodies that you see in this image. This is stained by a technique called the Golgi technique, and you can see now that beyond the cell body you see these branches that are coming from the cell body, and those are called dendrites. And the dendrites are the place where connections are made between different neurons. And so as the child matures over the first several years of life, the dendrites get more complicated because more synapses are formed on the dendrites, and ultimately a mature brain is developed. And again, this takes several years. And there's ongoing processes. It isn't the case that even at 6 years of life, when the brain is 95% of the adult size, that brain maturation is complete, because there are processes that continue even into the adolescence period that refines the organization of the brain. So it's a long-term process that goes all the way from fetal life into the first 2 decades of life. Now, when we think about autism and how to try and define the alterations in brain development that take place in autism, it's been a complicated process. In other disorders, such as Alzheimer's disease, we actually see markers of the disease in the brain. So this is a picture of Alzheimer, who first discovered in the early 1900s that when he observed microscopic sections through the brains of people who had had dementia, and ultimately that one form of dementia that he defined took on his name, Alzheimer's disease, that the neurons had unusual profiles in their cell bodies, what he called tangles. And you can see one of the neurons. This is a very high magnification a picture of a neuron, but the black object is the neurofibrillary tangle that he was able to define. And subsequent to Alzheimer's initial work, people have done tremendous amount of neuropathological assessment of the Alzheimer's brains. And now we know that somebody who has Alzheimer's form of dementia, their brains will always have tangles that you see in panels B here and D, and what are called amyloid plaques that you see in panels A and C. So that this is the hallmark of Alzheimer's disease and is what causes the degenerative process that ultimately leads to the dementia, loss of memory, and the other symptoms of Alzheimer's disease. But thus far, as people have looked for these kinds of markers in the brains of individuals with autism, we haven't been able to find those yet. But the reality is that there has not been all that much analysis of postmortem analysis of the brains from individuals with autism. So the first neuropathological studies of autism were carried out in the mid-1980s. And these studies were carried out initially by Margaret Bauman and Tom Kemper, who were in Boston, and looked at brains and found in qualitative studies where they made observations that there were some changes in the brains. They found patches of neurons that were too small and looked like they hadn't matured, but the number of brains that were evaluated were relatively few, on the order of 6 brains. And subsequently, there have been other investigators who've looked at the brains of individuals with autism. So folks like Jeff Hutzler, who's at the University of Nevada in Reno, has found that there are certain neurons in locations of the brain that seem like they have too many connections, too many synapses. But again, the number of brains that have been looked at have been relatively small. Manuel Casanova has also done studies in which he has found that the organization of the cerebral cortex, the basic columnar organization, or how neurons communicate with each other, looks to be altered, and has proposed that this is a fundamental problem in autism. And most recently, Eric Courchain and colleagues at the University of California, San Diego, have proposed that some brains of individuals with autism actually have too many neurons, particularly in the frontal lobe, and have proposed that perhaps this is part of the problem leading to abnormal behavior in autism. So as provocative as these studies are, the real problem in the field is that none of these studies have been replicated. And they've not been replicated not so much because there isn't interest or because there aren't investigators who would love to try and replicate these studies, but the problem has been that there is a critical shortage of postmortem brains for analysis. And in fact, through the entire history of autism research, there's been fewer than 150 brains that have been studied, and a very small proportion of those have been studied in a quantitative way. So there really is a a critical gap here in our understanding of the neuropathology, in part because we don't have the substrate to be able to study. Now, there are studies that use magnetic resonance imaging that indicate some issues related to the neuropathology of autism. So as Casey had said, we have a project going on here at the Mind Institute called the Autism Genome Project. And in this project, we have carried out MRI analysis of the brains of a large number of children, nearly 300 at this point in time. And what we're finding is that there are different patterns of abnormal brain development. So just like everybody appreciates that there's enormous heterogeneity in the behavioral characteristics and in the comorbid symptoms of autism, what we're finding is that there are different patterns of altered brain development as well. So as one example, it's been proposed for many years now that the brains of individuals with autism are too large. And in fact, when we have looked at the brains of the children in our study, and our study starts when the children are very young, so they're first evaluated when they're only 3 years of age. And it's very representative because children with all forms of autism, with all levels of severity, are involved in this project. And so it's sort of, I think, representative of what we will ultimately see in terms of neuropathology and autism. And what we find is that, sure enough, there is a group of children with autism who have large brains. So in this illustration, you can see that the child on the right has a substantially larger brain. This child with autism here, his brain is 20% larger than this child who also has autism at the same age. But it turns out that the percentage of children that have these enlarged brains is only about 15% of the boys. The vast majority of boys have brain size that is within the normal range, within the typical range. And interestingly, while we have imaged a smaller number of girls, our finding thus far is that this large-brained form of autism is not very common in girls. In fact, we found very few girls thus far that have what we call megalencephaly. So this would suggest that our task of understanding the neuropathology of autism is even harder, because we don't expect there to be one pathology like in Alzheimer's disease. We expect that there may actually be several forms of pathology. Now, you might say, well, why don't you just image a large number of children and solve the problem that way? And the reality is that magnetic resonance imaging has a resolution that's simply too low to see neurons and networks. So in this slide, you can see again a magnetic resonance image of a child. And what I've shown is that if you were to take just one single pixel, what we call a voxel from an MRI, it's about 1 cubic millimeter in size. That 1 cubic millimeter contains on the order of 50,000 neurons and 100 to 300 million synapses. So MRI, while it's valuable to give us the big picture of potential pathology in the brain, it certainly is not going to allow us to get down to the cells and the connections and the synaptic transmission problems that we think are going to be most valuable for us to move from research to more effective treatments. Now, it's not only in morphological studies that brains are needed for analysis, but it's also in the genetics of autism. And there are a number of recent studies over the last couple of years suggesting that if we're really going to understand psychiatric disorders, we are going to have to study the DNA from the brain and not simply DNA from the blood. So there are several examples, but here is a study that recently was published in Science really just within the last month or so, I believe, from the laboratory of Christopher Walsh at Children's Hospital in Boston. And what that group has shown is that perhaps because of the rapid turnover or rapid development of neurons during neurogenesis, there are mutations within the DNA in neurons that you don't see in the DNA from blood. And in fact, what these new studies are showing is that there may be mutations in single neurons that are different across across different regions in the brain. So this would suggest that if these mutations are influential on the alterations in the brain leading to autism, that we're never going to understand them by studying blood. We really do have to study brain. This is a topic that is coming up over and over again. There have been wonderful papers by Dan Geschwin and his colleagues at UCLA. They've shown that if you look at gene expression in the brain, that there's alterations in different portions of the brain in autism. In fact, there's less differential expression of genes in the brain in autism than in typical control specimens. Again, a very provocative study that has not, even though it was published a few years ago, has not been replicated because of lack of tissue. And there is very exciting work coming from the laboratory of Matt State and colleagues at University of California, San Francisco, again suggesting that the genes that are most linked or most highly linked to autism have a very strong and preferential expression in the brain during a particular portion of fetal life. And that area of research makes very strong hypotheses and predictions about where we will see the pathology in the autistic brain. But again, those predictions haven't been able to be looked at because of the lack of brains. So in the end, we have really reached a bottleneck. We have fantastic scientific techniques. We have highly motivated investigators who are ready and willing to explore some of these very exciting hypotheses about the causes of autism. This research could lead to new understanding of the neuropathology, the genetics, and neurochemistry of autism, which will probably these are the kinds of data that will most rapidly lead to new and effective treatments that will lead to decreases in disability in autism, yet we don't have the postmortem brain donations to support all of these forms of research. So we know that based on the MRI and genetic research that in order to foster this kind of research, we're going to need a large number of clinically and genetically well-characterized brains. We're going to also need, which is equally important, appropriate control brains for comparison. And this is going to come up again, that the kinds of donations we're looking for are not only from individuals who had autism during life, but also for donations of the brains of individuals who had typical development, so that we can do comparisons. And the point of this presentation today is that this has reached such a crisis that a few years back, my colleagues and I approached a number of organizations about the need to generate a new effort to bring this, to try and make the autism community aware of the need for postmortem brain donations. And I'm delighted that we were able to, it didn't take much convincing, it just took some organization, but we have developed what we call now Autism BrainNet. And I'd like to just tell you a little bit about this organization now that is attempting to solve the problem of the lack of postmortem brains for autism research. So Autism BrainNet is a collaborative effort It's sponsored by the Simons Foundation Autism Research Initiative. It's co-sponsored by Autism Speaks. And our outreach partner on this entire effort is Autism Science Foundation. This is an organization that has been in the works for at least a couple of years. It was formally announced in May of 2014, and it's still a work in progress, but let me tell you what we've set up so far. We decided that on a number of principles, and one is that we wanted the autism community to feel comfortable with the whole process of making a donation to Autism BrainNet. And so we thought that in part what would make families feel most comfortable if they would donate to an institution that was in their own region of the country. So Autism BrainNet has started with 4 locations, or we call them nodes, located in Sacramento, Dallas, Boston, and New York. So beyond being able to do local outreach to the community in these areas that are high population areas, the other reason for having a distributed network of collection sites is that for postmortem brains to be optimally useful for research, the time between the death of the individual and the proper preservation of the brain donation must be kept as short as possible. So up until recently, the length of time, which we call the postmortem interval, has been relatively long, 30 hours or longer. But to optimize the use of the tissue, we're aiming to bring this down to less than 24 hours, and what would be absolutely optimal is if the donation was made between 6 and 12 hours after death. Now, at each of these sites, we've established directors: Cindy Schumann in California, Carol Tominga in Texas, Matt Anderson in Boston, and Patrick Hoff in New York. And all of these individuals are either neuropathologists or physicians who have had a long history of collecting brain donations for research. And we have our first international colleague, Dr. Olaf Ansorge, who's the director of the Oxford Brain Bank in Great Britain. The principle under which this works is that regardless of whether a donation comes into the California node or the Boston node or the Texas node, that all of the information will go into a consolidated website. And then when researchers request tissue, that tissue will come from any of these locations. So the idea is that there will be a collaborative effort. All of the donations go into one collaborative pool, and then we will hope to provide tissue to as many researchers as possible through this mechanism. In this short webinar, I won't be able to give you all the details about Autism BrainNet, but we do have a website that's set up, and you can go to autismbrainnet.org and get many more details. We have news items on that website, and this website will continue to be updated as we get more information. You'll also see that in the top right here of this website, we have a 24/7 hotline. And we have a system at all of the nodes of clinicians who are ready and willing to take a call from a family that would like to make a donation. And what I can tell you is that we've spent an enormous amount of time to ensure that our clinicians are sensitive to the fact that the family is going through a traumatic period, and that we make the donation as easy and as sensitive as possible. So virtually all of the details are handled for the donation once consent is received, and the clinicians are very, very effective and very kind in the way they work with the families in making the donation happen. Now, I won't again go into the details of the organization of Autism BrainNet, but I did want to point out that we want the entire operation of Autism BrainNet to be as transparent as possible. So we have set up an organization where we can bring brain donations into our repositories at the different nodes. We have done everything we can to ensure the security of the brain donation, the safety of the brain donation for now and into the future, and then the decisions on who will receive brain tissue is handled by an independent scientific review committee. The scientific review committee is independent of the node directors, and the scientific review committee will evaluate applications from scientists. If the applications are scientifically meritorious and if tissue is available to support the request, tissue will then be sent to the investigators. And then we're making a strong plea that the investigators, once they've done their study, that the research data comes back to Autism BrainNet, and we will have a database which will then incorporate all of the data on all of the brain donations, again, to optimize the amount of information that we will get on the disorder. So while we now are fully functional, since 2014, we've had 14 brain donations. All of those brain donations have gone very smoothly. And we think at this point, the most important aspect of this whole effort is to bring the word about Autism BrainNet and the need for brain donations to the community. And this is why these kinds of webinars like we're doing today are so very important. And we're absolutely convinced that this effort will only be successful if the community is fully engaged and supportive, and in fact act as ambassadors for Autism BrainNet. And I would appreciate all of you who are on this call today to, if you're a researcher, to make your research subjects aware of Autism BrainNet. If you're an individual in the community, that you let other community members know about Autism BrainNet. And through the 877 number and through other contact information on our website, any additional questions that don't get answered today can be raised, and we'll certainly be happy to get back to folks with answers. But because outreach is so important, we've been delighted that Allison Singer, who's the president of Autism Science Foundation, has taken responsibility for the outreach efforts and has done really a remarkable job with her team, particularly Alicia Holliday, who is the Chief Scientific Officer for Autism Science Foundation, has worked very closely with the team from Autism BrainNet to begin the process of educating the community about the need for postmortem brains. In the early stages of this, Allison and the team did a series of focus groups, and they divided the focus groups into different categories. Group 1 were parents who had children who are 3 to 10 on the autism spectrum. Group 2, 11 to 17, and Group 3 who had older children with autism. And then there was a group that were adults with autism, all canvassed about their knowledge about the need for brain donations. And so this first panel shows that when she asked, after seeing the campaign, the It Takes Brains to Solve Autism campaign, what would people do? the responses were actually pretty favorable, that at least people would think about the issue or they'd seek more information about brain donation. A lot of positive responses. And what she realized after the initial surveys that she did was, in fact, there really wasn't that much awareness in the community that it is an essential need for brain donations to advance autism research. So you can see in these questions here that only 33% of people understood that the brain tissue of people with autism can be donated after death for use in autism research, or that the brain tissue of family members could be donated, and that you can help autism research by donating brain tissue from individuals who don't have autism but have typical development. So it was clear that we needed to do a lot more education of the community in general, that postmortem brain donations are essential for leading to an understanding of the causes of autism. This was an interesting finding that Allison discovered, and that is that branding is really, really important, and this is why it's critical that we have the major autism advocacy groups participating in the support of this effort. So these are people were asked whether they would consider donating brain tissue for autism research, and either their own brain tissue or the brain tissue of the child who has autism or the brain tissue of a child without autism. And there was reasonable support for that, so 51% donating their own tissue and somewhat less for children. But what Allison found was that when you let people know that this is an effort supported by the Simons Foundation and the Mind Institute, Autism Speaks, and Autism Science Foundation, people felt more comfortable with the possibility for donating, and all of these numbers that are positive are going up. And so, again, as you communicate to the community the need for postmortem brain tissue, I think it's important that you say that this is not simply an effort mounted by me or even by a small group of autism researchers, but by the major advocacy and funding groups in the United States. And so I think that there's a tremendous room for enlisting the help of people and for getting a larger number of sub-donations in the future. And when Allison asked whether you'd be willing or not willing to think or consider about a donation, what she found was that 55% of the people that she surveyed in her focus groups would be willing to consider donating, and the largest reason, of course, was to help other children and to support research. The people who were undecided really hadn't gotten enough information about the need for postmortem brains. But still, a very sizable proportion of the community, I think, with education, could perhaps go from the undecided into the would-be-willing category. And then there was a small proportion of individuals who are not willing to donate, and the reasons given were that either they considered it unethical or a very reasonable concern is that some individuals simply don't want to think about the topic of postmortem donations while their children are alive, and it's probably this group that will be more difficult to convince. But nonetheless, we would like to aim for those who would be willing or undecided and bring the message to them. So Allison has launched a campaign, an awareness campaign, and this is a campaign that at its heart is families talking to other families. So this was the very beginning of the campaign. The folks that you see in this picture are the Matthews family. They have one son with autism, and they live in the New York area, and these advertisements have gone out widely in the New York area. And Christine, the mom, has been a very strong ambassador for the Autism BrainNet and the TakesBrains initiative in the New York area. And the theme of the campaign is that families who have a child with autism are already heroes in providing the great support that they do to their affected family members. But if you go beyond that and consider the possibility of making a brain donation, then you become superheroes. And there's a little bit of humor in this with the capes and whatever. And Allison, again, as just one of the consummate a marketer, has realized that families don't want to understand or know the details of research necessarily. They don't want to see pictures of brains. They really want to hear about the benefits of autism research, which are decreasing disability and coming up with more effective treatments. And so the entire It Takes Brains to Solve Autism campaign is not so much about the process, but about the product, which will be better lives for people on the autism spectrum in the future due to autism research, or due to postmortem autism research. So there is a second website that is more directed at family members. This is at TakesBrains.org, TakesBrains.org, and on that website there's a Q&A region of the website that will tell you about the facts of postmortem brain donation. There's a location that talks about some of the research findings that have come out from postmortem brain research. And then most importantly, there's a sign-up region. And it says register to donate, and that's been a little confusing to some people. There's no obligation when individuals register for the TakesBrains registry. What this simply does is allow us to provide families and individuals more information about autism research that is supported by postmortem donations. So it really is signing up for a quarterly newsletter, again, that Alicia Holliday has taken responsibility in generating, and provide more information to families that they could find useful in considering ultimately making a donation or passing along information to other community members who would consider making a donation. So, and I would like to ask you today to consider signing up. Again, there's no obligation. It does not obligate you to make a donation, but it will provide you with additional information on an ongoing basis about the Autism BrainNet and It Takes Brains efforts. On the website, as I said, there's a question and answers location, and there are family-friendly questions and answers. Why should my family consider donation, for example? And it goes through religious and ethical components. It talks about why other techniques other than postmortem brain research can't answer the questions. And again, it provides the information that you would need to have to consider and to communicate the need for postmortem donations to your community. Just another example, what have scientists learned, what do they hope to learn, again, provides basic information about why we're doing this. And then again, there's the sign-up page, which I mentioned, which the sign-up page, when you get to that page and you click register to donate, there's a 30-second registration page that simply asks you to provide name and some contact information. Literally takes just about 30 seconds to do. And what that will allow us to do will be to send the quarterly newsletter, and again, provide a conduit for you to communicate back to us. We really want advice from the community about how this should proceed, and we want to establish a relationship with the community to consider making donations. Now, one of the things that we're doing is we're taking this campaign to the community. So Alicia and several volunteers have been going to a variety of Autism Speaks walks, for example, and other kinds of functions in which a portion of the autism community is available. And again, they provide information about It Takes Brains and Autism BrainNet. They usually have iPads so that they can sign folks up for the newsletter, and this is going to be an ongoing basis. There's a very large population of families affected by autism in the United States, and we've really just begun to scratch the surface in getting the message out to families. And again, this is why these kinds of webinars are so important, but we can't do it alone. We really need your help to bring the message to the community. If, for example, we have a 5x7 card that we distribute at these events, if there are people listening to this webinar who are members of community groups or other organizations where you would like to have some of these 5x7 cards to distribute to your group, please contact us, and we'd be happy to send the cards to you and make it as easy as possible for you to help us spread the word about Autism BrainNet. Again, it only takes a few minutes for you to sign up, so don't wait. You can do it during the question and answer period or immediately after the webinar. Please go on to TakesBrains.org, register for the newsletter, and we'll be able to continue having a discussion with you about this topic. So I just wanted to mention that in my last slide to a couple of current efforts where we're doing outreach. We've developed a collaboration with the Dup15q Alliance. Dup15q is a genetic modification that often leads to autism, and this advocacy group has teamed up with Autism BrainNet so that they are making their community aware that if they would like to make a donation, that Autism BrainNet would support the collection of donations from this community. We would like to reach out to all of the different sub-communities of individuals with autism and develop partnerships with these communities. And then on our website, we will have a partners page that will not only indicate that we have formed the partnerships, but again, talk in detail about the specific contribution that Autism BrainNet has made to understanding the specific form of autism. So Dup15q Alliance was the first, but we're hoping for many more partners like this. And one thing that we're hopefully in the very final stages of is negotiating with the NIH Neurobiobank. So the National Institute of Health has its own effort to collect postmortem brains. In fact, they've realized that nationally we don't have enough brain donations for not only autism but other disorders as well. So they have reinvigorated their brain bank system, and they call it NIH Neurobiobank. And when they did this, we realized that there could be a perception that maybe this is a competition for Autism BrainNet, and we didn't want that to be the case. So we have been in negotiations for about a year with the National Institute of Health about the idea of, again, collaborating on the collection of donations from individuals with autism nationally to develop one global outreach that would have both a public, the NIH, and a private component, Autism BrainNet. But then we come to terms so that the donations are collected using the standard operating procedures, and that distribution could be done collaboratively. We're not quite there yet, but I'm hoping that in the very near future we'll be able to make an announcement that we have come to agreement on doing one global collaborative collection of autism brains throughout the United States. And then finally, we are going to increase our efforts to bring the message to families, and particularly to medical examiners around the country, because the sad reality is that there are many more deaths of individuals who are on the autism spectrum than brain donations are coming from. And we think in large part this is because the message hasn't gotten out that there is a really severe need for these kinds of donations to advance autism research. And for the next year or so, at the very least, we're going to try and intensify our efforts to bring this message to families and to medical examiners who are seeing individuals who are potential donors. So again, I'm very appreciative to the Simon Simplex and Consortium and to Ian for allowing me to present this overview of Autism BrainNet and It Takes Brains, and I'm happy to try and answer as many questions as we can in the remaining time. Thanks very much. Casey, I'll turn it back to you.
Thanks so much, Dr. Amaral. It's a really important topic, and we appreciate your time as well and discussing this with us in the community. We do have a number of questions that have come in from the audience. There are some of them that are sort of, I guess, process or specific to It Takes Brains. I'll start with those and then move into some of the research questions. So there are some folks who are curious about organ donation versus brain donation, and whether if you're an organ donor, you can also donate your brain, or if that happens automatically, or what the difference is there.
Right. So it's a very good question. If you are an organ donor, you can also do a brain donation, but it is not typically a part of the donation process for organ donations. So for people who have already taken the step of agreeing to being an organ donor, they would also have to take the step of informing their next of kin, for example, that they would like their brain donated to Autism BrainNet. It doesn't happen automatically. You would have to indicate that you'd like that to be the case. So organ donation does not necessarily mean that the brain will be donated to research.
Okay, thanks. There are a few folks just wondering about the general success of Autism BrainNet as far as how many donations have been received since it was established. Was that the 14 that you spoke about since May 2014?
That's right. There were 14 since May of last year. We have, because this is a collaboration with Autism Speaks, the brains that remained within the Autism Tissue Program in Autism Speaks have now become part of the collection for Autism BrainNet. So there were at least portions of some brains that were donated previous to the establishment of Autism BrainNet that have come into our collection as well. We have 14 new brains that have been donated since the launch. And I should say that there were an additional 11 donations that came into the UC Davis Mind Institute Bears Brain Bank, and those will become part of our collection as well. So still a very small number, but the number seems to be increasing, and we have been getting increased numbers of calls as, again, people become more aware of the need.
Sure. And so related to that, and I know that this question does not have an easy or standardized answer, but folks are wondering, what is the typical number of brains needed to publish a paper, for example, using the postmortem tissue?
Right, and that isn't an easy question to answer. It really depends on which type of science you're looking at. So the number can be as few as less than 10 if you're looking at a genetic form of autism and there's more homogeneity of that form of autism. If you're trying to do a histological study, then it's probably on the order of 20 or 30 brains or more. And if you're doing a wide-scale genetic study, then the larger the number, the better. Again, the problem is that we're dealing with a very heterogeneous disorder that has multiple causes. And the reality is, for all research on autism, but particularly for the biological features of autism, the larger the number of cases that you have to study, the more likely you are to be able to make sense out of the situation. So that, again, is one of the reasons why, for us really to understand the brain pathology or the brain genetic pathology, we're attempting to get the very largest number of brains possible.
Sure. One last sort of procedural question. We have a number of SSC families who are in Canada, and they're wondering what their options are, if this is just in the U.S. and then Britain right now.
Yes. So at the moment, one of our nodes in the United States is able to accept donations from Canada. In fact, one of the 14 brain donations that I mentioned during the last year came from Ottawa. So our Canadian colleagues and community can simply call the 877 number, and we can facilitate the collection of donations from Canada as well. We are hoping to have a Canadian node in the not too distant future, and we have begun discussions with some potential directors both on the east coast and west coast of Canada. Those haven't been formalized yet, but again, if a family in Canada would like to make a donation, we can accept those donations now through one of our other nodes.
Okay, perfect. I would say that we have about 2 or 3 minutes left. The bulk of the rest of the questions that have come in relate to exactly what brain research can tell us about autism. So some of these questions are things of the nature, can you study autism by separating the brain from the body when autism is really a behavior disorder, that kind of thing. So what kinds of questions can brain research really help us to address that relate directly to treatment and daily living diagnosis type issues, which I know is a really wide question, but in a few minutes, is it possible to summarize some of the areas of research?
Sure. Well, let me just give you an example of a disorder where it was brain research that has led to the various kinds of treatments. In Parkinson's disease, which is a motor impairment of aging and also leads to cognitive dysfunctions, until really the middle part of the last century, we didn't understand what caused it, and ultimately we realized through research and research with postmortem brains that it's a small group of neurons that have a particular transmitter called dopamine that seem to degenerate in Parkinson's disease. And once we understood that those were the cause for the disabilities associated with Parkinson's disease, we were able to understand the other parts of the brain that were affected, and this led directly to the development of the treatments using dopamine or L-DOPA and some of the other treatments that are currently being used, even deep brain stimulation and others. For autism, we are still struggling to understand the fundamental biological problems that take place, whether it's the alteration of a specific, it looks like there's a large amount of circumstantial evidence, primarily from the genetic research that's going on, that there are problems at the synapse, at communication at the synapse, but what we haven't been able to figure out is whether it's the specific types of synapses, whether it's a specific neurotransmitter that's most impacted, and which parts of the brain are most impacted. Postmortem brain research is what is going to lead us to an understanding of or an answer to all those questions. And once we answer those questions, that will immediately lead to potential therapies that will ameliorate those communication problems in those portions of the brain. So until we understand the basic biology, we don't really have a target for treatment. Postmortem brain research will ultimately give us several targets that will then be able to attempt to find pharmacological treatments for.
All right, yeah, such a complicated problem, but certainly this is an important topic to help us to address some of the issues that the field's most interested in taking a look at. So I appreciate it, once again your time, Dr. Amaral. I think we really are out of time as far as our Q&A session for today. So I thank everybody also in the audience for attending, and just a reminder that we will post the recording of the webinar on the IAN website, the Interactive Autism Network website. It should go up within a week or two of today's date. So as soon as we get it, you will be able to find it there, and you can view it, send it out, and we'll also let everyone know the date for our next SSC at IAN webinar, which will be in early 2016. You'll receive information about that as well through our newsletters. And so thank you once again, Dr. Amaral, for your time. We really appreciate you speaking with us today about this topic, and we hope everyone has a wonderful day.